Hi, I'm Madeline. I'm a PhD student here at the Burnet.
Hello, my name is Thorey and I'm also a PhD student here at the Burnet in malaria research.
Today we're going to give you a behind-the-scenes tour of what we do here in the labs.
So come on, join us and see what our daily life looks like here at the Burnet Institute.
The malaria parasite exports hundreds of parasite proteins to modify the red blood cell and make it into a livable habitat. I am characterizing few of these proteins, and to do this, I genetically modify the parasite's DNA, which involves a process called polymerase chain reaction, or PCR for short. Here you can see me loading my final PCR product, which contains multiple copies of my DNA of interest on a gel. I have added a blue loading dye to my DNA samples, which adds density to the DNA, so it falls into the loading wells. The gel also has a DNA stain added to it to allow me to visualize the DNA bands after the gel has run by exposing the gel to UV light. The gel itself contains small pores, and we push the DNA across the gel using electrical field, where large fragments take longer to go through the gel than the smaller ones. The DNA itself is negatively charged and runs towards a positively charged electrode. To make sure that the gel is running, we always check if small bubbles run towards the surface after starting the run, which indicates that the electrical current is present. The plates you see here are multicycle growth assays for some of my genetically modified parasite lines. These assays help me determine if the protein I am studying is essential for parasite survival. I have incorporated a genetic switch to my proteins, which allow me to basically switch off the protein and then monitor how that affects the parasite growth. We are interested in identifying proteins essential for parasite survival because they could be excellent drug targets.
Thorey has just shown you a parasite growth experiment, and this is where we grow the parasites. They live in big incubators that keep them nice and warm at 37 degrees to match the temperature that they would be in the human body. We are here today in one of the instrument rooms in the labs at the Burnet Institute, and this is a special microscope that we can use for our experiments. These parasites are very, very small. If you think about how tiny a grain of rice is, these parasites are more than 1,000 times smaller. So with this microscope, we can watch and film these tiny parasites to see how they are behaving. The part of the parasite that we're interested in is how they get out of the red blood cell and get into new red blood cells. The reason we are interested in this is because we are trying to find new medicines to stop them from being able to do this, and this would treat people who get sick with malaria. The special part of this microscope is that it's a humidified microscope, which means it keeps the parasites warm and happy, replicating what it's like in the human body, like we saw in the incubator room. So these are some of the type of videos we can see when we do these experiments. We look for red blood cells that have parasites in them and wait for them to burst from their red blood cells. When it looks like the parasites are ready to burst, we start filming them. You can see in the videos that all the larger circle shapes are red blood cells, and the little tiny dots are the parasites that are all looking for a new red blood cell to make their home in. What's so useful about this technique is that we can re-watch the videos in a much slower speed than it happens in real life, which allows us to really look closely at what the parasites are doing. Another experiment we can do using this microscope to study the parasite is to use special colored dyes. You can see that here the red blood cell is stained green while the little parasite is stained red. And we can see using these dyes how the parasite gets into the red blood cell so we can understand this process better.
Thank you for watching us here today. I hope our work has inspired you to take up a career in research.
We look forward to seeing you here one day.